Introduction. The human body contains approximately 37 trillion cells, with bacterial cells outnumbering the eukaryotic cells by about 10 to 1. Our cells create tissues that form organs, and each cell serves a purpose, communicating and carrying out the reactions that make life possible. The sheer number of cells that make up the human body is not nearly as impressive as the numerous functions these cells can perform, from the conduction of impulses through the nervous system that allows for memory and learning, to the simultaneous contraction of cardiac myocytes that pump blood through the entire human body. In order to understand the human organism as a whole and how the human body reacts to various pathogens, a thorough understanding of cell biology is required. It is not enough to simply memorize each part of the cell. The MCAT requires an understanding of how each cell structure carries out its functions and affects the entire organism. 1.1 Cell theory. Prior to the 1600s, organisms were perceived as being complete and inseparable into smaller parts. This was due in part to the inability to see smaller structures through simple optical instruments like magnifying glasses. In 1665, Robert Hooke assembled a crude compound microscope and tested its properties on a piece of cork. He noticed a honeycomb-like structure and compared the spaces within the cork to the small rooms of a monastery, known as cells. Because cork consists of desiccated non-living cells, Hooke was not able to see nuclei, organelles, or cell membranes. In 1674, Anton van Leeuwenhoek was the first to view a living cell under a microscope. Later researchers noted that cells could be separated, and that each cell was a distinct structure. Further research indicated that tissues were made of cells, and the function of a tissue was dependent upon the function of the cells that make up the tissue. 2 centuries later, in 1850, Rudolf Virchow demonstrated that diseased cells could arise from normal cells in normal tissues. The original form of the cell theory consisted of 3 basic tenets: all living things are composed of cells. The cell is the basic functional unit of life. Cells arise only from pre-existing cells. Through advances in molecular biology, a 4th tenet has been added to the theory: cells carry genetic information in the form of deoxyribonucleic acid, DNA. This genetic material is passed on from parent to daughter cell. Cell theory has created an interesting dilemma with respect to viruses. Viruses are small structures that contain genetic material, but are unable to reproduce on their own. This violates the 3rd and 4th tenets of the cell theory because virions can only replicate by invading other organisms and because they may use ribonucleic acid, RNA, as their genetic information. Therefore, viruses, discussed later in this chapter, are not considered living organisms. (1.2) The first major distinction we can make between living organisms is whether they are composed of prokaryotic or eukaryotic cells. Prokaryotic organisms are always single-celled, while eukaryotic organisms can be unicellular or multicellular. Whereas eukaryotic cells contain a true nucleus enclosed in a membrane, prokaryotic cells do not contain a nucleus. The major organelles are identified in the eukaryotic cell. Membrane-bound organelles. Each cell has a membrane enclosing a semi-fluid cytosol in which the organelles are suspended. In eukaryotic cells, most organelles are membrane-bound, allowing for compartmentalization of functions. Membranes of eukaryotic cells consist of a phospholipid bilayer. This membrane is unique in that its surfaces are hydrophilic, electrostatically interacting with the aqueous environments inside and outside of the cell, while its inner portion is hydrophobic, which helps to provide a highly selective barrier between the interior of the cell and the external environment. The cell membrane is such an important topic on the MCAT that an entire chapter 8 of MCAT Biochemistry Review is devoted solely to discussing the structure and physiology of biological membranes. The cytosol allows for the diffusion of molecules throughout the cell. Within the nucleus, genetic material is encoded in deoxyribonucleic acid, DNA, which is organized into chromosomes. Eukaryotic cells reproduce by mitosis, allowing for the formation of two identical daughter cells. The nucleus as the control center of the cell. The nucleus is the most heavily tested organelle on the MCAT. It contains all of the genetic material necessary for replication of the cell. The nucleus is surrounded by the nuclear membrane or envelope, a double membrane that maintains a nuclear environment separate and distinct from the cytoplasm. Nuclear pores in the nuclear membrane allow selective two-way exchange of material between the cytoplasm and the nucleus. The genetic material, DNA, contains coding regions called genes. Linear DNA is wound around organizing proteins known as histones, and is then further wound into linear strands called chromosomes. The location of DNA in the nucleus permits the compartmentalization of DNA transcription separate from RNA translation. Finally, there is a subsection of the nucleus known as the nucleolus, where the ribosomal RNA, rRNA, is synthesized. The nucleolus actually takes up approximately 25% of the volume of the entire nucleus and can often be identified as a darker spot in the nucleus. Mitochondria. Mitochondria are often called the power plants of the cell, in reference to their important metabolic functions. The mitochondrion contains two layers, the outer and inner membranes. The outer membrane serves as a barrier between the cytosol and the inner environment of the mitochondrion. The inner membrane, which is arranged into numerous enfoldings called cristae, contains the molecules and enzymes of the electron transport chain. The cristae are highly convoluted structures that increase the surface area available for electron transport chain enzymes. The space between the inner and outer membranes is called the intermembrane space. The space inside the inner membrane is called the mitochondrial matrix. As described in Chapter 10 of MCAT Biochemistry Review, the pumping of protons from the mitochondrial matrix to the intermembrane space establishes the proton motive force. Ultimately, these protons flow through ADP synthase to generate ADP during oxidative phosphorylation. Mitochondria are different from other parts of the cell in that they are semi-autonomous. They contain some of their own genes and replicate independently of the nucleus via binary fission. As such, they are paradigmatic examples of cytoplasmic or extranuclear inheritance, the transmission of genetic material independent of the nucleus. Mitochondria are thought to have originated when the engulfing of an aerobic prokaryote by an anaerobic prokaryote resulted in a symbiotic relationship. In addition to keeping the cell alive by providing energy, the mitochondria are also capable of killing the cell by release of enzymes from the electron transport chain. This release kickstarts a process known as apoptosis, or programmed cell death. Lysosomes. Lysosomes are membrane-bound structures containing hydrolytic enzymes that are capable of breaking down many different substrates, including substances ingested by endocytosis and cellular waste products. Lysosomes often function in conjunction with endosomes, which transport, package and sort cell material traveling to and from the membrane. Endosomes are capable of transporting materials to the transgolgi, to the cell membrane, or to the lysosomal pathway for degradation. The lysosomal membrane sequesters these enzymes to prevent damage to the cell. However, release of these enzymes can occur in a process known as autolysis. Like mitochondria, when lysosomes release their hydrolytic enzymes, it results in apoptosis. In this case, the released enzymes directly lead to the degradation of cellular components. Endoplasmic reticulum. The endoplasmic reticulum, ER, is a series of interconnected membranes that are actually contiguous with the nuclear envelope. The double membrane of the endoplasmic reticulum is folded into numerous invaginations, creating complex structures with a central lumen. There are two varieties of ER, smooth and rough. The rough ER, rare, is studded with ribosomes, which permit the translation of proteins destined for secretion directly into its lumen. On the other hand, the smooth ER, SER, lacks ribosomes and is utilized primarily for lipid synthesis, such as the phospholipids in the cell membrane, and the detoxification of certain drugs and poisons. The cere also transports proteins from the rare to the Golgi apparatus. Golgi apparatus. The Golgi apparatus consists of stacked membrane-bound sacs. Materials from the ER are transferred to the Golgi apparatus in vesicles. Once inside the Golgi apparatus, these cellular products may be modified by the addition of groups like carbohydrates, phosphates, and sulfates. The Golgi apparatus may also modify cellular products through the introduction of signal sequences, which direct the delivery of the product to a specific cellular location. After modification and sorting in the Golgi apparatus, cellular products are repackaged in vesicles, which are then directed to the correct cellular location. If the product is destined for secretion, the secretory vesicle merges with the cell membrane and its contents are released via exocytosis. Peroxisomes. Peroxisomes contain hydrogen peroxide. One of the primary functions of peroxisomes is the breakdown of very long-chain fatty acids via beta-oxidation. Peroxisomes participate in the synthesis of phospholipids and contain some of the enzymes involved in the pentose phosphate pathway, discussed in Chapter 9 of MCAT Biochemistry Review. The cytoskeleton. The cytoskeleton provided structure to the cell and helps it to maintain its shape. In addition, the cytoskeleton provides a conduit for the transport of materials around the cell. There are three components of the cytoskeleton: microfilaments, microtubules, and intermediate filaments. Microfilaments. Microfilaments are made up of solid polymerized rods of actin. The actin filaments are organized into bundles and networks and are resistant to both compression and fracture, providing protection for the cell. Actin filaments can also use ADP to generate force for movement by interacting with myosin, such as in muscle contraction. Microfilaments also play a role in cytokinesis, or the division of materials between daughter cells. During mitosis, the cleavage furrow is formed from microfilaments, which organize as a ring at the site of division between the two new daughter cells. As the actin filaments within this ring contract, the ring becomes smaller, eventually pinching off the connection between the two daughter cells. Microtubules. Unlike microfilaments, microtubules are hollow polymers of tubulin proteins. Microtubules radiate throughout the cell, providing the primary pathways along which motor proteins like kinesin and dynein carry vesicles. Cilia and flagella are motile structures composed of microtubules. Cilia are projections from a cell that are primarily involved in the movement of materials along the surface of the cell. For example, cilia line the respiratory tract and are involved in the movement of mucus. Flagella are structures involved in the movement of the cell itself, such as the movement of sperm cells through the reproductive tract. Cilia and flagella share the same structure, composed of 9 pairs of microtubules forming an outer ring, with 2 microtubules in the center. This is known as a 9+2 structure and is seen only in eukaryotic organelles of motility. Bacterial flagella have a different structure with a different chemical composition, as discussed later in this chapter. Centrioles are found in a region of the cell called the centrosome. They are the organizing centers for microtubules and are structured as 9 triplets of microtubules with a hollow center. During mitosis, the centrioles migrate to opposite poles of the dividing cell and organize the mitotic spindle. The microtubules emanating from the centrioles attach to the chromosomes via complexes called kinetochores and exert force on the sister chromatids, pulling them apart. Intermediate filaments. Intermediate filaments are a diverse group of filamentous proteins, which includes keratin, desmin, vimentin, and lamins. Many intermediate filaments are involved in cell-cell adhesion or maintenance of the overall integrity of the cytoskeleton. Intermediate filaments are able to withstand a tremendous amount of tension, increasing the structural rigidity of the cell. In addition, intermediate filaments help anchor other organelles, including the nucleus. The identity of the intermediate filament proteins within a cell is specific to the cell and tissue type. Tissue formation. One of the unique characteristics of eukaryotic cells is the formation of tissues. Division of labor, as different cells in a tissue may carry out different functions. For example, in the heart, some cells participate in the conduction pathways while others cause contraction, still others serve a supportive role like maintaining structural integrity of the organ. There are 4 tissue types: epithelial tissue, connective tissue, muscle, and nervous tissue. While muscle and nervous tissue are considered more extensively in subsequent chapters, we explore epithelial and connective tissues below. Epithelial tissue. Epithelial tissues cover the body and line its cavities, providing a means for protection against pathogen invasion and desiccation. In certain organs, epithelial cells are involved in absorption, secretion, and sensation. To remain as one cohesive unit, epithelial cells are tightly joined to each other and to an underlying layer of connective tissue known as the basement membrane. Epithelial cells are highly diverse and serve numerous functions depending on the identity of the organ in which they are found. In most organs, epithelial cells constitute the parenchyma or the functional parts of the organ. For example, nephrons in the kidney are epithelial cells, and hepatocytes in the liver, and acid-producing cells of the stomach are epithelial cells. Epithelial cells are often polarized, meaning that one side faces a lumen, the hollow inside of an organ or tube, or the outside world, while the other side interacts with underlying blood vessels and structural cells. For example, in the small intestine, one side of the cell will be involved in absorption of nutrients from the lumen, while the other side will be involved in releasing those nutrients into circulation for use in the rest of the body. We can classify different epithelia according to the number of layers they have and the shape of their cells. Simple epithelia have one layer of cells, stratified epithelia have multiple layers, and pseudostratified epithelia appear to have multiple layers due to differences in cell height but are, in reality, only one layer. Tuning to shape, cells may be classified as cuboidal, columnar, or squamous, as their names imply. Cuboidal cells are cube-shaped and columnar cells are long and thin. Squamous cells are flat and scale-like. Connective tissue supports the body and provides a framework for the epithelial cells to carry out their functions. Whereas epithelial cells contribute to the chyma of an organ, connective tissues are the main contributors to the stroma or support structure. Bone, cartilage, tendons, ligaments, adipose tissue, and blood are all examples of connective tissues. Most cells in connective tissues produce and secrete materials such as collagen and elastin to form the extracellular matrix. 1.3 Classification and structure of prokaryotic cells. Prokaryotes are the simplest of all organisms and include all bacteria. Prokaryotes do not contain any membrane-bound organelles, and their genetic material is organized into a single circular molecule of DNA concentrated in an area of the cell called the nucleoid region. Despite the simplicity of prokaryotes, they are incredibly diverse, and knowledge of this diversity is essential for the study of medicine because many prokaryotes can cause infection. In fact, choosing the appropriate antibiotic to fight an infection requires knowledge about the basic structure of the bacteria causing the infection. Prokaryotic domains. There are 3 overarching domains into which all life is classified: archaea, bacteria, and eukarya. 2 of these, archaea and bacteria, contain prokaryotes. Initially, archaea and bacteria were classified together into the kingdom of Munera. However, modern genetics and biochemical techniques have indicated that the differences in the evolutionary pathways between archaea and bacteria are at least as significant as between either of these domains and eukarya. Archaea are single-celled organisms that are visually similar to bacteria, but contain genes and several metabolic pathways that are more similar to eukaryotes than to bacteria. Historically, archaea were considered extremophiles, in that they were most commonly isolated from harsh environments with extremely high temperatures, high salinity, or no light. More recent research has demonstrated a greater variety of habitats for these organisms, including the human body. Archaea are notable for their ability to use alternative sources of energy. While some are photosynthetic, many are chemosynthetic and can generate energy from inorganic compounds, including sulfur and nitrogen-based compounds such as ammonia. Due to the similarities of this domain to eukaryotes, it is hypothesized that eukaryotes and the archaea share a common origin. Both eukaryotes and archaea start translation with methionine, contain similar RNA polymerases, and associate their DNA with histones. However, archaea contain a single circular chromosome, divide by binary fission or budding, and share a similar overall structure to bacteria. Interestingly, archaea are resistant to many antibiotics. All bacteria contain a cell membrane and cytoplasm, and some have flagella or fimbriae, similar to cilia. Because bacteria and eukaryotes often share analogous structures, it can be difficult to develop medicines that target only bacteria. However, in some cases, even seemingly similar structures have enough biochemical differences to allow the exclusive targeting of one kind of organism. For example, bacterial flagella and eukaryotic flagella are different enough that scientists are able to develop antibacterial vaccines that specifically target the bacterial flagellum. Also, many antibiotics target the bacterial ribosome, which is significantly smaller than the eukaryotic ribosome. There are approximately 5x 1030 bacteria on Earth, outnumbering all of the outnumber human cells in the body by 10:1. The relationship between the hum and animals combined. As mentioned in the introduction to this chapter, bacteria body and bacteria is complex. Some bacteria are mutualistic symbiotes, means that both humans and the bacteria benefit from the relationship. Examples include bacteria in the human gut that produce vitamin K and biotin, vitamin, and which also prevent the overgrowth of harmful bacteria. Other bacteria are pathogens parasites, meaning that they provide no advantage or benefit to the host, but rather cause disease. Pathogenic bacteria may live intracellularly or extracellularly. For example, Chlamydia trachomatis, a common sexually transmitted infection, lives inside cells of the reproductive tract. Clostridium tetani, the cause of tetanus, lives outside of cells and produces toxins that enter the bloodstream. Classification of bacteria by shape. Classification of bacteria by shape provides scientists and pathologists, physicians who specialize in the identification and characterization of disease, a common language to talk about bacteria, as well as a way to identify different species of bacteria. Most bacteria exist in one of three shapes. Spherical bacteria, known as cocci, include common pathogens such as Streptococcus pyogenes. Rod-shaped bacteria, like Escherichia coli, are known as bacilli. Finally, spiral-shaped bacteria, known as spirili, include such species such as Treponema pallidum, which causes syphilis. Aerobes and anaerobes. Some bacteria require oxygen for survival, while others do not. Bacteria that require oxygen for metabolism are termed obligate aerobes. Other bacteria that use fermentation, or some other form of cellular metabolism that does not require oxygen, called anaerobes. There are different types of anaerobes. Anaerobes that can survive in an oxygen-containing environment are called obligate anaerobes. Presence of oxygen leads to the production of reactive oxygen-containing radicals I. These species, which leads to cell death. Other bacteria can toggle between metabolic processes, using oxygen for aerobic metabolism if it is present, and switching anaerobic metabolism if it is not. These bacteria are called facultative anaerobes. Finally, aerotolerant anaerobes are unable to use oxygen for metabolism, but are not harmed by its presence in the environment. Prokaryotic cell structure. One of the main differences between prokaryotes and eukaryotes is that prokaryotes lack a nucleus and membrane-bound organelles. Prokaryotes are also single-celled organisms, meaning that each cell must be able to perform all of the functions necessary for life on its own. However, prokaryotes may live in colonies with other cells and may signal these cells to share information about the environment. Cell wall. Because prokaryotes do not form multicellular organisms, each bacterium responsible for protecting itself from the environment. The cell wall forms the outer barrier of the cell. The next layer is the cell membrane, plasma membrane, which is composed of phospholipids, similar to that of a eukaryote. Together, the cell wall and the cell membrane are known as the envelope. The cell wall both provides structure and controls the movement of solutes into and out of the bacterium. This allows the cell to maintain a concentration gradient relative to the environment. In bacteria, there are two main types of cell wall: gram-positive and gram-negative. The type of cell wall is determined by the gram staining process, a crystal violet stain, followed by a counterstain with its substance called safranine. If the envelope absorbs the crystal violet stain, it will appear deep purple, and the cell is said to be gram-positive. If the envelope does not absorb the crystal violet stain, but absorbs the safranine counterstain, then the cell will appear pink-red, and the cell is said to be gram-negative. Gram-positive cell walls consist of a thick layer of peptidoglycan, a polymeric substance made from amino acids and sugars. In addition to its structural and barrier functions, the cell wall may also aid a bacterial pathogen by providing protection from a host organism's immune system. In addition to peptidoglycan, the gram-positive cell wall also contains lipoteichoic acid. It is not clear what role this acid serves for the bacterium, but the human immune system may be activated by exposure to these chemicals. Gram-negative cell walls are very thin and also contain peptidoglycan, but in much smaller amounts. The peptidoglycan cell walls of these bacteria are adjacent to the cell membrane, and are separated from the membrane by the periplasmic space. In addition to the cell wall and cell membrane, gram-negative bacteria also have outer membranes containing phospholipids and lipopolysaccharides. Interestingly, lipopolysaccharides are the part of gram-negative bacteria that triggers an immune response in human beings. The inflammatory response to lipopolysaccharides is much stronger than the response to lipoteichoic acid. Flagella. Flagella are long, whip-like structures that can be used for propulsion. Bacteria may have one, two, or many flagella, depending on the species. Flagella can be used to move toward food or away from toxins or immune cells. This ability of a cell to detect chemical stimuli and move toward or away from them is called chemotaxis. The flagella are composed of a filament, a basal body, and a hook, as shown in Figure 1.9. The filament is a hollow, helical structure composed of flagellin. The basal body is a complex structure that anchors the flagellum to the cytoplasmic membrane and is also the motor of the flagellum, which rotates at rates up to 300 Hz. The hook connects the filament and the basal body so that, as the basal body rotates, it exerts torque on the filament, which thereby spins and propels the bacterium forward. The overall structure of flagella is similar in both gram-positive and gram-negative bacteria, but there are slight differences due to the different physical structure and chemical composition of the envelope in gram-positive and gram-negative bacteria. Archaea also contain flagella, but the structure of their flagella is quite different from that of bacteria and is unlikely to be asked about on test day. Other organelles. As mentioned earlier, prokaryotes concentrate DNA in a region of the cell known as the nucleoid region, which does not contain a nuclear envelope. Prokaryotic DNA is carried on a single circular chromosome which can be found coiled around histone-like proteins in some bacteria. True histones are found in archaea. In addition, DNA acquired from external sources may also be carried on smaller circular structures known as plasmids. Plasmids carry DNA that is not necessary for survival of the prokaryote and therefore is not considered part of the genome of the bacterium but may confer an advantage such as antibiotic resistance. Prokaryotes lack several key organelles, such as mitochondria. Instead, the cell membrane is used for the electron transport chain and generation of ADP. Prokaryotes do contain a primitive cytoskeleton, but it is not neatly as complex as the one found in eukaryotes. Prokaryotes also contain ribosomes, but this ribosome is a different size from that found in eukaryotes. Prokaryotic ribosomes contain 30s and 50s subunits, whereas eukaryotic ribosomes contain 40s and 60s ribosomes. (1.4) Genetics and growth of prokaryotic cells. As we have seen, prokaryotic cells differ from eukaryotic cells both structurally and biochemically. Prokaryotes reproduce via asexual reproduction in the form of beneficiation. In addition, prokaryotes are capable of acquiring and using genetic material from outside the cell. Binary fission is a simple form of asexual reproduction seen in prokaryotes. The circular chromosome attaches to the cell wall and replicates while the cell continues to grow in size. Eventually, the plasma membrane and cell wall begin to grow inward along the midline of the cell to produce two identical daughter cells. Because binary fission requires fewer events than mitosis, it can proceed more rapidly. In fact, some strains of E. coli can replicate every 20 minutes under ideal growth conditions. Genetic recombination is necessary for the cell to survive and reproduce. However, many bacteria also contain extrachromosomal, extragenomic, material known as plasmids. Plasmids often carry genes that impart some benefit to the bacterium, such as antibiotic resistance. Some mechanisms. Plasmids may also carry the single circular chromosome of a prokaryotic cell contains the information that additional virulence factors, or traits that increase pathogenicity, such as toxin production, projections that allow attachment to certain kinds of cells, or features that allow evasion of the host's immune system. A subset of plasmids called apisomes are capable of integrating into the genome of the bacterium. Bacterial genetic recombination helps increase bacterial diversity and thus permits evolution of a bacterial species over time. These recombination processes include transformation, conjugation, and transduction. Transformation results from the integration of foreign genetic material into the host genome. This foreign genetic material most frequently comes from other bacteria that, upon lysing, spill their contents into the vicinity of a bacterium capable of transformation. Many gram-negative rods are able to carry out this process. Conjugation. Conjugation is the bacterial form of mating, sexual reproduction. It involves two cells forming a conjugation bridge between them that facilitates the transfer of genetic material. The transfer is unidirectional, from the donor male, plus, to the recipient female. The bridge is made from appendages called sex pili that are found on the donor male. To form the pilus, bacteria must contain plasmids known as sex factors that contain the necessary genes. The best studied sex factor is the F. fertility factor in E. coli. Bacteria possessing this plasmid are termed FN cells, those without are called F cells. During conjugation, the FN cell replicates its F factor and donates the copy to the F cell, converting it to an FN cell. This enables the cell obtaining the new plasmid to then transfer copies to other cells. This method of genetic recombination allows for rapid acquisition of antibiotic resistance or virulence factors throughout a colony because other plasmids can also be passed through the conjugation bridge. The sex factor is a plasmid, but through processes such as transformation, it can become integrated into the host genome. In this case, when conjugation occurs, the entire genome replicates because it now contains the sex factor. The donor cell will then attempt to transfer an entire copy of its genome into the recipient. However, the bridge usually breaks before the full DNA sequence can be moved. Cells that have undergone this change are referred to by the abbreviation HFR for high frequency of recombination. Transduction. Transduction is the only genetic recombination process that requires a vector, a virus that carries genetic material from one bacterium to another. Viruses are obligate intracellular pathogens, which means that they cannot reproduce outside of a host cell. Because of this, bacteriophages, viruses that infect bacteria, can accidentally incorporate a segment of host DNA during assembly. When the bacteriophage infects another bacterium, it can release this trapped DNA into the new host cell. This transferred DNA can then integrate into the genome, giving the new host additional genes. Transposons. Transposons are genetic elements capable of inserting and removing themselves from the genome. This phenomenon is not limited to prokaryotes, it has been seen in eukaryotes as well. If a transposon is inserted within a coding region of gene, that gene may be disrupted. Growth. As discussed previously, bacteria reproduce via binary fission. This implies that all of the bacteria are exactly the same in a local colony, assuming no mutations or genetic recombination, and that no bacteria are dividing faster than the others. Bacteria can be said to grow in a series of phases. In a new environment, bacteria first adapt to the new local conditions during the lag phase. As the bacteria adapt, the rate of division increases, causing an exponential increase in the number of bacteria in the colony during the exponential phase, also called the log phase. As the number of bacteria in the colony grows, resources are often reduced. The reduction of resources slows reproduction, and the stationary phase results. After the bacteria have exceeded the ability of the environment to support the number of bacteria, a death phase occurs, marking the depletion of resources. 1.5 Viruses and subviral particles. At the beginning of this chapter, we discussed the cell theory and noted that viruses do not fit the definition of living things because they are acellular. Viruses may be small as 20 nm or as large as 300 nm. For reference, prokaryotes are 1 to 10 nm and eukaryotes are about 10 times larger. Unlike eukaryotic cells, viruses lack organelles and a nucleus. Viral structure. Viruses are composed of genetic material, a protein coat, and sometimes an envelope containing lipids. The genetic information may be circular or linear, single or double-stranded, and composed of either DNA or RNA. The protein coat is known as a capsid, which may be surrounded by an envelope composed of phospholipids and virus-specific proteins. The envelope is very sensitive to heat, detergents, and desiccation, thus, enveloped viruses are easier to kill. On the other hand, viruses that do not have an envelope are more resistant to sterilization and are likely to persist on surfaces for an extended period of time. Because viruses cannot reproduce independently, they are considered obligate intracellular parasites. Viruses must express and replicate genetic information within host cell because they lack ribosomes to carry out protein synthesis. After hijacking a cell's machinery, a virus will replicate and produce viral progeny, called virions, which can be released to infect additional cells. Bacteriophages are viruses that specifically target bacteria. They do not actually enter bacteria, rather, they simply inject their genetic material, leaving the remaining structures outside the infected cell. In addition to a capsid, bacteriophages contain a tail sheath and tail fibers. The tail sheath can act like a syringe, injecting genetic material into a bacterium. The tail fibers help the bacteriophage recognize and connect to the correct host cell. Viral genomes. Viral genomes come in a variety of shapes and sizes. Some are made of only a few genes, while others have several hundred. In addition, viral genomes may be made of either single, or double-stranded DNA or RNA. Single-stranded RNA viruses may be positive sense or negative sense. Positive sense implies that the genome may be directly translated to functional proteins by the ribosomes of the host cell, just like mRNA. Negative sense RNA viruses are a bit more complicated. The negative sense RNA strand acts as a template for synthesis of a complementary strand, which can then be used as a template for protein synthesis. Negative sense RNA viruses must carry an RNA replicase in the virion to ensure that the complementary strand is synthesized. Retroviruses are enveloped, single-stranded RNA viruses in the family Retroviridae. Usually, the virion contains two identical RNA molecules. These viruses carry an enzyme known as reverse transcriptase, which synthesizes DNA from single-stranded RNA. The DNA then integrates into the host cell genome, where it is replicated and transcribed as if it were the host cell's own DNA. This is a clever mechanism because the integration of the genetic material into the host cell genome allows the cell to be infected indefinitely, so the only way to remove the infection is to kill the infected cell. The human immunodeficiency virus, HIV, is a retrovirus that utilizes this life cycle, which is one of the characteristics that make HIV so difficult to treat. Viral life cycle. As we have already discussed, viruses must infect a host cell and use the host cell's machinery in order to reproduce. Here, we will discuss the viral life cycle in detail. Infection. Viruses can only infect a specific set of cells. Without the proper receptors, a cell is essentially invisible to the virus. Once the virus binds the correct receptor, the virus and the cell receptors on the host cell. Without the proper receptors, a cell is essentially invisible to the virus. Once the virus binds the correct receptor, the virus and the cell are brought into close enough proximity to permit additional interactions. Enveloped viruses fuse with the plasma membrane of a cell, allowing entry of the virion into the host cell. Sometimes a host cell may mistake a virus bound to the membrane as nutrients or other useful molecules and will actually bring the virus into the host bacterium through the tail sheath. Some tail fibers even have enzymatic activity, allowing for both penetration of the cell wall and the formation of into the cytoplasm via endocytosis. As mentioned earlier, bacteriophages use tail fibers to anchor themselves to the cell membrane and then inject their genome pores in the cell membrane. Depending on the virus, different portions of the virion will be inserted into host cells. Enveloped viruses such as HIV fuse with the membrane and enter the cell intact, whereas bacteriophages only insert their genetic material, leaving their capsids outside the host cell. Translation and progeny assembly. After infection, translation of viral genetic material must occur in order for the virus to reproduce. This requires translocation of the genetic material to the correct location in the cell. Most DNA viruses must enter the nucleus in order to be transcribed into mRNA. The mRNA then goes to the cytoplasm, where it is translated into proteins. Genetic material from positive sense RNA viruses stays in the cytoplasm, where it is directly translated into protein by host cell ribosomes. Negative sense RNA viruses require synthesis of a complementary RNA strand via RNA replicase, which can then be translated to form proteins. DNA formed through reverse transcription in retroviruses also travels to the nucleus, where it can be integrated into the host genome. Using the ribosomes, tRNA, amino acids, and enzymes of the host cell, viral RNA is translated into protein. Many of these proteins are structural capsid proteins and allow for the creation of new virions in the cytoplasm in the host cell. Once the viral genome has been replicated, it can be packaged within the capsid. Note that the viral genome must be returned to its original form before packaging. For example, retroviruses must transcribe new copies of their single-stranded RNA from the DNA that entered the host genome. A single virus may create anywhere from hundreds to many thousands of new virions within a single host cell. Progeny release. Viral progeny may be released in multiple ways. First, the viral invasion may initiate cell death, which results in spilling of the viral progeny. Second, the host cell may lyse as a result of being filled with extremely large numbers of virions. Lysis is actually a disadvantage for the virus because the virus can no longer use the cell to carry out its life cycle. Finally, a virus can leave the cell by fusing, which this process keeps the host cell alive, and thus allows for the continued use of host cell by the virus. Lytic and lysogenic cycles. Depending on growth conditions and the specific virus, bacteriophages may enter a lytic or lysogenic life cycle. These two phases are similar to the lysis and productive cycle methods of progeny release discussed above. During a lytic cycle, the bacteriophage maximizes the use of the cell's machinery with little regard for the survival of the host cell. Once the host is swollen with new virions, the cell lyses, and other bacteria can be infected. Viruses in the lytic phase are termed virulent. In the event that the virus does not lyse the bacterium, it may integrate into the host genome as a provirus or prophage, initiating the lysogenic cycle. In this case, the virus will be replicated as the bacterium reproduces because it is now a part of the host's genome. Although the virus may remain integrated into the host genome indefinitely, environmental factors, radiation, light, or chemicals, may cause the provirus to leave the genome and revert to a lytic cycle. As mentioned earlier, the provirus may extract bacterial genes as it leaves the allows transduction of genes from one bacterium to another. Although bacteriogenome, which phages can kill their hosts, integration of the phage into the host genome may actually benefit the bacterium. Infection with one strain of phage generally makes the bacterium less susceptible to superinfection, simultaneous infection, with other phages. Because the provirus is relatively innocuous, this arrangement may confer an evolutionary advantage. Prions and viroids. Prions and viroids are very small, subviral, particles that can cause disease under certain circumstances. Prions. Prions are infectious proteins and are, therefore, non-living things. They cause disease by triggering misfolding of other proteins, usually through the conversion of a protein from an OE helical structure to a SS pleated sheet. This drastically reduces the solubility of the protein, as well as the ability of the cell to degrade the protein. Eventually, protein aggregates form, interfering with cell function. Prions are known to cause bovine spongiform encephalopathy, mad cow disease, Creutzfeldt-Jakob disease, and familial fatal insomnia in humans. Viroids. Viroids are small pathogens consisting of a very short circular single-stranded sequences RNA that infect plants. Viroids can bind to a large number of RNA sequences and can silence genes in the plant genome. This prevents synthesis of necessary proteins, resulting in metabolic and structural damage cell. Viroids are classically thought of as plant pathogens, but a few examples of human viroids do exist, including the hepatitis D virus, HDV. Alone, HDV is innocuous, however, when co-infected with hepatitis B virus, HBV, HDV is able to exert its silencing effect on human hepatocytes. Conclusion. Our first chapter introduced the basis of all biology, cell theory. All living things are made of prokaryotic or eukaryotic cells. Prokaryotes are simpler and do not contain membrane-bound organelles. Eukaryotes contain membrane-bound organelles with highly specialized functions. While eukaryotic organisms, especially humans, will be the primary focus on test day, an understanding of prokaryotic structure and physiology is necessary to understand infectious disease. Viruses are non-living infectious particles that must invade cells in order to reproduce. Finally, we discussed the smallest infectious particles, prions, and viroids. Our discussions from here on out will focus primarily on eukaryotes, but you will spend significant time in your clinical career battling the pathogens we've presented in this chapter. Vaccines are available for a number of bacteria: Bacillus anthracis anthrax, Corynebacterium diphtheriae diphtheria, Haemophilus influenza type, many upper respiratory and ear infections, Neisseria meningitidis, some cases of bacterial meningitis, Streptococcus pneumoniae, many cases of bacterial pneumonia, Clostridium tetani tetanus, Salmonella typhi typhoid, and Bordetella pertussis whooping cough, and viruses, Varicella zoster virus, chickenpox and shingles, hepatitis A and B viruses, human papillomavirus HPV, influenza, measles, mumps, polio, rabies, rotavirus, rubella, and yellow fever. Those for which we do not have vaccines may be targeted with antibiotic and antiviral therapies. The appropriate pharmacotherapy usually depends on an understanding of bacterial or viral physiology and the known resistance patterns in the local area. In other words, the principles presented in this chapter will show up in your everyday life as a physician. We've discussed one method of cellular reproduction, the division of a bacterial cell into two cells by binary fission. Eukaryotic cells also must replicate, but use a different process, mitosis. In the next chapter, we will turn our attention to mitosis, as well as meiosis and human reproduction.